चैप्टर का नाम है डीएनए फिंगरप्रिंटिंग। It is a technique to identify person on the basis of his या her DNA specifically. It is invented by Sir Elick Jeffrey 1984 में किया था। It involves identifying different some specific region DNA sequence and sequence called repetitive DNA. These repetitive DNA are separated from bulk genomic DNA as different peaks during density gradient configuration. The bulk DNA forms major peak of the other small peaks are referred to satellite DNA. The satellite DNA is classified into many categories such as mini satellite, microsatellite, etc. This sequence shows the high degree of polymorphism and from the basis of DNA fragment. और पॉइंट है mini satellites are 1160 bp long repetitive sequence where number and position is variable each chromosomes and also person to person, so they are known as VNTR, variable number of tandem repeats. VNTR in a DNA is a flanked by restriction site and known as restricted fragment. The length of restricted fragment of each chromosome DNA variable and this known as RFLP, restricted fragment length polymorphism. The fingerprinting techniques is based on RFLP. The DNA fingerprinting techniques include following steps: isolation DNA, digestion of DNA restriction into cleavage, separation of DNA fragment by electrophoresis, transforming of separate DNA fragments of systematic membrane such as nitrocellulose or nylon southern bolting. Hybridization use label VNTR probe detected hybridization fragment by auto radiography. The auto radiography gives mainly bands of different size. It differs from the individual to individual in a population except in a case of monozygotic twins. This technique is very useful in forensic science, paternity testing, in determining population and genetic diversity. Cloning. Formation of clones is cloning. Clone is group of genetically identical organisms. Asexual or vegetative reproduction produce natural clones. Clone can be done artificially. It may be gene cloning, microbial, cell cloning, plant cloning, or animal cloning. Animals cloning difficult and not direct as plant because animal cellulose loses potency in early embryonic stage. Animal cloning may be reproductive cloning, therapy cloning. In reproductive cloning of whole animal is developed, it have there three techniques: blastomer separation techniques, nuclear transplantation techniques, Honolulu technique. In therapeutic cloning, only desired organs are produced from cell plant patient. This is a very useful medical science. Dolly sheep are the produced using nuclear transfer technique by Dr.